Hello boys. So again, I'm just going over the last couple of things we did with the genetic crosses over the last couple of weeks. Some of you missed out. Even if you were in class, no harm to watch over this and just have a quick look over it again. So in this video, we're going to look at two types of linkage, gene linkage and sex linkage. So first of all, gene linkage. And this is when genes are located on the same chromosome. And the impact of this is that if the genes are on the same chromosome, the alleles cannot sort independently, as the linked alleles must pass into the gametes together. So therefore, Mendel's second law does not occur. There is no independent assortment. So let's look at two crosses here. One where we consider the genes are not linked, and then we'll do this exact same cross where the genes are linked. So here we have, this is the same as I was looking at in the other video. It's the pea plants, and we have the round yellow dominant over the wrinkled green. So in this case here, we have the first parent is heterozygous dominant, the other parent is homozygous recessive. Now, for this, I'm going to draw what we call a chromosome diagram, just showing little lines to show the chromosomes. So in this case, the genes are not linked. So the genes for seed shape are on different chromosomes to the genes for seed color. Okay, so we see here we have both heterozygous for both of the traits, and here we have homozygous recessive for both of the traits. So when we look at the gametes, so we could have the capital R with the capital Y, or the capital R with the small y, small r with the small y, or the small r with the capital Y, or the small r with the small y. In this case here, we only have one option, so it has to be a small r with a small y. So we see there's the four options from the first parent, one option from the second parent, and just to do a Punnett square to illustrate this. So we put the gametes of one parent along the top, gametes of the other parent on the side, and we match them up, and we see four possible outcomes of genotypes here, and each one of them is different. And we look at the phenotypes. So the first one there, that's round yellow. Second one is round green. Third one, wrinkled yellow, and then wrinkled green. And so they're in the 25% chance of either of these, or they're in the ratio of 1:1:1:1. Okay, now, so the offspring show all possible combinations of the traits in equal proportions. So let's go and look at the situation now where the genes are linked. Now, and again, we have a chromosome diagram to show this. So same parents, heterozygous and homozygous recessive. So we look in this case now, the genes are linked. So the gene for seed shape is on the same chromosome as the gene for seed color. Same over here. Now, so what's the impact of that? Well, as I said earlier, independent assortment cannot occur, because in this case, the capital R and the capital Y are on the same chromosome. So during gamete formation, the two of these stay together, and the small r must stay with the small y. So they're the only two possible combinations we can get in the gametes in this situation, as opposed to remind ourselves up here, when linkage did not occur, we could get four possible combinations. So back down to this. Again here, there's only one combination like before, and there's no Punnett square here, so the capital R, capital Y cross with the small r, small y. And there's the heterozygous offspring, or the small r, small y will cross with small r, small y to give the homozygous recessive. So we notice that as a result of the linkage, there is less variety in the offspring, as opposed to having four different types of offspring. We now only have 2 different types of offspring. And if you go back, just up a little bit here, they are the same as the parents. Now, it's important to note that this is the only type of cross you can be asked about in gene linkage, where one parent is heterozygous, the other is homozygous recessive. So, for example, the cross in the other video where both parents are heterozygous, you cannot be asked that in gene linkage. Okay, this is the only type you need to know. And often they'll ask, they will say linkage does not occur or linkage does occur, and they will ask then, do your cross and state the impact of the gene linkage. So first of all, in terms of gamete formation, the impact is, to remind us on that, as we said up here, the alleles do not sort independently, as the linked alleles must pass together into the gametes. And also then the impact on the offspring, there's less variety in the offspring. The offspring are the same as the parents, as opposed to showing all possible combinations that we saw when the genes were not linked. Okay, moving on now to what we call sex linkage. So this is where genes are on the sex chromosomes. So there are very few genes on the X chromosome because the X chromosome is shorter. Now, since males have only one X chromosome, all genes on the X chromosome that have no corresponding gene on the Y, whether dominant or recessive, they are expressed. Okay? In contrast, a mutant gene, often disease-causing, on an X chromosome in a female is usually covered up by the normal allele on the other X chromosome. We'll look at an example of that. So in this case, we're looking at sex-linked conditions, sex-linked diseases. And the disease is caused by a mutant gene. Now, because it's a mutant gene, it tends to be quite rare. So in females, if a female does have the mutant gene, it's normally covered up or masked by the normal dominant allele, so the female doesn't suffer. Now, however, as we'll see later on, if the male has this mutant gene, it is going to suffer from the condition. Right, so again we look at an example here. Here are 2 conditions caused by sex-linked genes. Red-green color blindness and hemophilia. So hemophilia is a condition where people find it difficult for their blood to clot. It doesn't clot very well, so they have huge issues around bleeding when they get a cut. So the example here, so the gene for color vision is sex-linked, and the allele for normal vision, capital N, is dominant to the allele for color blindness, small n. So what would the genotype and phenotype of the offspring be when a carrier mother, so she's a carrier, that means she has the allele for color blindness, but this is masked by the dominant normal allele. So, and she's having children with a normal father. So the father has the normal gene. Okay, so again, we're going to use chromosome diagrams throughout this. And for these sex-linked conditions, you only really use the chromosome diagrams. So here we see the mother. She's a carrier. She has the normal dominant allele along with the recessive. So she does not suffer from the condition because the normal dominant allele masks the recessive. The father then, so remember, he's only one allele for this trait, because the Y chromosome is shorter. There's no matching allele on the Y chromosome, and he has the dominant allele. Right. So here we look at the gamete formation. Again, going with the chromosome diagrams. So here we have in the female. So these are the possible female eggs. One has the allele for the dominant normal allele. The other has the recessive colorblind allele. Whereas the father then, we see here, he has the in the sperm that he forms, half of them have the normal allele, the other half just have the Y chromosome. Right, so then we look at how these match up in fertilization as the gametes fuse together. So we could have this egg being fertilized by this sperm, and that's the case there, with the genotype capital N, and the phenotype. Now it's important on the phenotype, give the gender as well. So this is a normal girl. And then we have the situation where, again, that egg is fertilized by the other sperm. That's the sperm with the Y chromosome. So we have a normal boy. Now, here we have the situation then where the egg with the recessive allele fertilizes. First of all, with the sperm with the normal allele. And there, in this case, we have a carrier girl. And in the other case, where this egg is fertilized by the sperm with the Y chromosome, we're going to have a color-blind boy. Now, if you notice in this, it's only the boys that can be colorblind in this situation. The girls are either normal, they're homozygous dominant, or they are carriers. Okay? But we see, as I mentioned earlier, if a boy has the recessive allele, it will suffer from the condition because it cannot have a dominant allele to mask that. So the Y chromosome has no gene for the trait, so males only need to inherit one recessive allele from their mother for them to suffer from the disease. They cannot have a dominant allele to mask that recessive allele. Females must inherit the recessive allele from both parents. As a result, males are more likely to suffer from sex-linked diseases. Okay, so that's a question that could be asked. Why are males more likely? And we say because females must inherit the recessive allele for both parents, or you can say that the male only needs to inherit one recessive allele, and it's always from their mother they inherit that from, for them to suffer from the condition. And the very, very final little thing here is this notion of non-nuclear inheritance. So what we're looking at here is parts of the cell that contain DNA apart from the nucleus. So and we have two examples of this, and these are the chloroplasts and the mitochondria. Okay? Now, just an interesting note about the mitochondria. If you remember when we talked about fertilization, when the sperm fertilizes the egg, only the head of the sperm enters the egg. So therefore, the mitochondria in the collar, or at the midpiece of the sperm, they do not enter the egg. So the mitochondria of the sperm, which originated from the father, that stays outside the egg. So all the mitochondria in the zygote comes from the mother. Okay? So all the DNA in an individual's mitochondria came from that person's mother. None of the DNA in the mitochondria comes from the father. Okay, so that's just an interesting point to remember there on that. Right, okay, thanks very much.